Welcome to our next lecture. Today we're going to be talking about the seedless vascular plants, still part of chapter 29. And the goal for today is to get through kind of what seedless vascular plants are, how are they similar to the bryophytes that we talked about last time, and then obviously what are some of the derived traits that we see appear in this group. We have two representative phyla that you'll need to know. The first phylum is called Lycophyta, and then the second is called Monolophyta. Okay, so the bryophytes, guys, they were around for a long time. They were quite successful for reasons that I discussed in the previous lecture, and they basically ruled the landscape for about 100 million years. So if you think about that, they were very successful and kind of spoiled for a long time because they didn't have really any other plants to compete with. But over time, as you know, the bryophytes then are not the only land plants that we have, and we have plants that are able to get much larger in size and derive even more traits that we don't see in the bryophyte group. So if you can imagine having mats of bryophytes like carpet, right? Growing and everything's about, let's just say, 1 centimeter tall, and you have a mutation in a plant and it gets to be about 1.5 centimeters tall. Well, that's going to be able to outcompete all of the other bryophytes simply because there's going to be competition for light. And so evolutionary adaptations allow for plants to get much larger, and I'm talking 6 feet by 240 feet, right? So huge plants eventually evolve, and that's going to be due to competition for light and space. As those two competitive factors increase, then the plants had to do everything that they could to survive, and therefore we end up with something that looks like this. Okay, so this is a very swampy forest filled with many seedless vascular plants. They actually end up causing their own demise, many of them, which I'll talk about at the end of the lecture. So just kind of in your mind right now, guys, just kind of remember what this picture looks like. Okay, so this is a concept map that shows the plants, and I thought it was very useful. Might be something that you guys want to be able to draw out as you're studying the group of plants here, Kingdom Plantae. So we covered this last time, the non-vascular plants. But now we're going to go over here to our vascular plants, and we're talking about seedless vascular plants. So these are plants that do not have any seeds. That'll be the topic for the next lecture. And we have examples then listed here under seedless vascular plants, and those are going to be ferns, horsetails, and club mosses. So I'll go through those plants with you, some of which I'm pretty sure that you're going to be familiar with. Okay, so just a quick rundown on what these guys here, right, what they have as far as shared derived characteristics. So if I put the word shared in front of it, that means that they were not the first group to evolve the traits, but they've been passed from previous ancestors. In this case, the bryophytes are passing then these traits on to seedless vascular plants. And that should make sense, right? Because if a trait is working, then it should be passed on. It should not be eliminated from the population. So if we're looking at our notes, I'm on page 8, and I'm under A1, part A. So all of the seedless vascular plants, they maintain their cuticle. Alternation of generations is still the way that they reproduce. They still have stomata, and they are still dependent upon water for reproduction. And so hopefully by now you realize that means that they need water because their sperm cells are flagellated. Okay, and then one other thing I would like for you to add in your notes under D, I forgot to add this in here, is that these plants are still spore producing. So that means that they reproduce via spores, which is their reproductive cell, because they do not have seeds. So spore reproducing is a term we can use for the seedless vascular plants, our topic for today, and then also for the non-vascular plants that we talked about last time. Okay, so I'm going to go ahead and go over some of the differences then, some of the derived traits that we see appear in this group of seedless vascular plants. And we're going to start with A, the dominant generation. So I'll come to this slide in a second. The dominant generation here in alternation of generations life cycle is the sporophyte stage. So the sporophyte stage is the one that is dominant. So for example, if I were to ask you what stage of the life cycle is this here, because you're looking at a plant that has vascular tissue, you should be able to tell me that that is in fact the sporophyte stage. Remember that dominant means that that's the one that is completely independent and it can photosynthesize on its own. One of the evolutionary trends that I'm going to continue to talk about over time is the fact that the gametophyte is going to get smaller and smaller and smaller. And so here, for this group of seedless vascular plants, the gametophyte is reduced to sometimes only being a couple of millimeters, usually right under the layer of the soil, and something that you would never see. So it's basically the complete opposite of what we talked about with the bryophytes. So again, the dominant stage of the life cycle here is the sporophyte stage. Okay, one of the other derived traits is as the sporophyte then gets to be larger and becomes an independent entity, it also appears then to have this dichotomous branching. So if you see here, every kind of stem, if you will, branches into two, thus dichotomous branching. And you can see that that's over here in this picture as well. And the dichotomous branching is going to allow then for the production of more sporangia. So each one of these kind of little orange nebby things at the end of each branch is a single sporangium. Sporangia is plural. And you can see them labeled here. This is going to be from figure 29.11 in your textbook. And the sporangium is essentially a holder of spores, right? Sporangium. So if this is a holder of spores, making spores, then you should automatically know at this point that this is on the sporophyte stage of the life cycle. And if I were to ask you, this is a great quiz question, tell me what the ploidy level is of all of the tissue here. And because it's sporophyte, you should be able to tell me that that is diploid. If I were to ask you what is produced inside these sporangia here, your answer should be spores. And then if I were to ask you what is the process then by which spores are made, you should be able to tell me that that is meiosis. Because if you remember, if you go back to your alternation of generations life cycle, right, your spores are haploid. And I just mentioned that this tissue is diploid. So meiosis is occurring inside of the sporangia then to make lots and lots of haploid spores. And what's going to happen is, because this plant is able to get taller for reasons that I'll talk about in a second compared to your mosses, and we have branching here, you get increased sporangia, you get increased spores. You get increased spores, that's going to give you more gametophyte. If you get more gametophyte, right, you get greater numbers of gametes. Greater number of gametes gives you greater number of zygotes. Zygotes then develop into increased sporangia. So the cycle is able to produce more of itself due to the increased number of sporangia. And then we need to not overlook the fact that because these are higher off of the ground, these sporangia here, they're able to release their spores and increase then their basically their departure from the parent plant, because they have a higher likelihood of being carried then by wind currents because they're up off of the ground. So this is just a really good thing for the plant to increase then its reproductive capacity. Okay, so in your notes 2B, part 1, we're going to talk about spore production. So I just mentioned that, just kind of went through that quickly there as a reminder then from the alternation of generations life cycle. But one thing that I want to point out is that plants are either going to be homosporous or heterosporous. So what I'm talking about here is what type of spores is the plant making inside of its sporangium. So if the plant is described as being a homosporous plant, if we look at the prefix here, right, homo means the same. If the plant is a heterosporous plant, or heterosporous, it's kind of a potato thing. Hetero means different. So if it's a homosporous plant, then that means the sporophyte, right, is going to make a single type of spore. That single type of spore makes a bisexual gametophyte, because there's only one gametophyte that can come from a spore. That bisexual gametophyte is going to have archegonia making eggs and antheridia making sperm. Okay, so you can't tell the difference based on what spore type if it's going to make an egg or a sperm, because they're both going to be made on the same gametophyte. Things are a little bit different down here. So we won't talk about any plants today that are heterosporous. They come next time, but I just wanted to define the term for you because it is in your book. If it is a heterosporous plant, the sporophyte then has two different sporangia. It has a megasporangium and a microsporangium. These terms are not on the slide, but they are in your notes. So once again, a sporophyte would make a megasporangium. The megasporangium would make a megaspore. The megaspore would develop into a female gametophyte. So this process here would be mitosis. The female gametophyte then, as you know, would be the archegonia, which would be busy making eggs via mitosis. The sporophyte also has a microsporangium to make microspores, which makes the male gametophyte, which would make the sperm cells. And this right here would be the antheridium, and as you know, the antheridium makes the sperm cells. So two different spores, a big spore and a small spore coming from two different sporangium, a megaspore and a microspore. And guys, this shouldn't be too confusing because if you think about in animals, right, something that you're used to, sperm and egg cells, which one is bigger? It's always going to be the egg cell. And the sperm cells are always going to be a fraction of the size of the sperm cell. I'm sorry, let me back up. The sperm cell is going to be a fraction of the size of the egg cell. So if you think mega, you already know that the egg is always bigger than the sperm, so that should help. And micro, you always know that the sperm is going to be much smaller than the egg. So this over here is an example of a plant that we will be looking at today. I'll be talking about this phylum here in a little bit, and this is also going to be something you'll be considering in the lab. But if we look at this, this kind of cone-like structure at the top, which I'll talk about that later, that's going to have the sporangia. Okay, so that's the term right here. And inside the sporangia are going to be spores, and those spores are going to be all of the same type. Those spores are released. They go through, they make a gametophyte. So this gametophyte you can see is very small. It has a very short life, basically has enough minerals and sugar in it just to survive long enough to make both egg and sperm. So this is an example of the bisexual gametophyte. Okay, and then again, water is required because the sperm cells are flagellated and they have to go swim to the egg in order for fertilization to occur, which is what is going to happen here. And then the life cycle starts all over again. So two things. This is showing then a single type of spore giving rise to a bisexual gametophyte, which means that this is a homosporous plant. And again, all of your plants today will be homosporous. Secondly, I want to point out that as we go through our life cycles for the groups here, these are all just different variations of alternation of generation. And not a variation in the process, but a variation in which stage is dominant. In this case, it's the sporophyte. Which stage is not dominant? That would be your gametophyte here, which is again the opposite of the bryophytes, okay? But all of the structures, even though they have the same name and they are doing the same thing, they look different. And that's why they're, again, in different groups. So this is just simply alternation of generations, and all they've done is they've applied then the different parts and the part stages to the life cycle. That is it. Okay, now what has allowed then these plants to get taller? That's going to be the stuff here called vascular tissues. So if we step back for just a second, what maybe the determining factor that allowed then mosses to actually make it to living on land were their gametangia. The gametangia, archegonia, and antheridia, right? The ability to make gametes and then also to protect them and to not just release them out into the water and hope for the best. Okay, so that was a huge derived trait that probably was most important, if you will, in leading to the success of the bryophytes. Here, even though they maintain their gametangia, this idea of vascular tissues, right? This is the feature that allowed plants to dominate the landscape. Again, getting to reach 6 feet tall, 6 feet in width and 240 feet tall. Think of a sequoia tree. That's a modern day plant that you might be familiar with. And so some of these seedless vascular plants were just as tall as your modern day sequoias. And that's going to be because of vascular tissue. Okay, so think of the vascular system and these vascular tissues, if you will, kind of as a plumbing system of the plant. The xylem right here and the phloem here, right? These are two different types of tissues that work together. And you could think of when they're together, that's the plumbing system, and each type of tissue then are the respective pipes, if you will. Okay, so we have xylem, and what xylem does is it moves in a one-way flow, and what it's going to move throughout the plant then, and you can see that it's going to go from the bottom up, so hopefully that should be a clue. It's going to be moving water and mineral nutrients that the roots pick up from the soil. Think of it as a really, really long kind of straw, if you will, because eventually these cells that make the tissue, the cell walls here, they all disintegrate, and so there's no walls then that are separating the individual cells upon maturity of this tissue in the xylem. We have really thick areas around the xylem. The stuff is called lignin, and this is going to eventually allow for woody types of plants to come about. But think about this, how thick it is on both sides. You're able to move water up 240 feet if that's where it needs to go, which is going to give great strength to the plant, allowing then for them to reach above 1 centimeter in height. Okay, working in conjunction with the xylem then is this stuff over here called phloem. And phloem, the movement, guys, of the photosynthates, so that's going to be things like sugars, amino acids, and the like that the plant is making for itself, is going to be moved throughout the phloem in the plant in a manner that is referred to as from source to sink. Source to sink. So that's basically a two-way flow. So source to sink means that the photosynthates are going to be moved from where they are made to where they are needed. Okay, so you've got a lot more flexibility here. You can tell that one of the differences, besides just the movement and the type of movement in the direction, is that each of these cells making up the phloem then, they actually are separated by cell walls. But if you look closely, these little teeny tiny green dots that are there, right? You can see them there and you can see them here and here. They kind of act as like sieve plates. And what that's going to allow for then, these are little tiny holes that are going to control the movement of what needs to go up or down from source to sink within the phloem. And then I've included this figure over here to the right, just so that you can see that the water is going to move up, and when water needs to move across, it can do that into the phloem, okay? But it can't go down. So it can go up, and it can be moved from the phloem, either from the xylem to the phloem or from the phloem to the xylem. But basically what I want you to take away from this is that this is just a one-way flow of water, but do appreciate that the xylem and the phloem, they do have to work together. This is showing then the movement of sucrose, which is a sugar that the plant is able to make on its own. So the sugar is made from is made at the source, i.e. in the leaf, and then it's going to be moved through the phloem to where it is needed. That's the sink, and that would be, for example, a root. So all cells in the roots, all cells in the stems and the trunk and the leaves, the very top of the tip of the tallest leaf, have to have water, and they have to have the photosynthates. So all of these tissues here are working together to make a vascular system kind of like yours, except that it's not moving blood. Okay, so just think of these kind of as maybe veins, if you will, that are responsible for moving specific things throughout the plant from top to bottom, and only in one direction in this case, and multi-directions in that case. Okay, these guys are going to be packaged together in what's called a vascular bundle. So that's going to be the next term here, see part 3 in the notes. So this here, you can kind of make out this orange layer of cells, very kind of thin layer of cells right here. And then you've got this kind of blue tissue and this red. So this red here is the xylem, and this blue tissue that's kind of in between the X's here, that's the phloem. And so these are phloem tubes and xylem tubes or tissues that are packaged together in what is called a vascular bundle. If we come and we look at this picture over here, this is going to show you several vascular bundles. So there's one, there's another one, there's another one, there's another one. And you can see they're just scattered throughout the entire section of whatever part of the plant this was that they used to make the slide. So one of these is expanded to look like one of these. Now, if we were in lab, face-to-face, what I would be telling you is that when you looked under the scope and you're looking at your vascular bundles, the coloring that they use to stain the tissues is always consistent. So that's nice because it helps you to be able to learn and remember what's going on. But within a vascular bundle, regardless of what it looks like, because they don't all take on the same exact pattern, as you'll see as we move through the rest of the plants. But nevertheless, the tissue that is inside the vascular bundle that has a red, sometimes it might be pink, that's always going to be xylem. And in this case, you can tell that the xylem cells are quite large, right? They're really big tubes allowing for water to move up. All of the kind of bluish green tissue, here, 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 and here, cells are much smaller. That's always going to be phloem. So again, in a vascular bundle, when you're making your drawings, or if I pop this onto the next quiz or whatever, and I ask, what is this tissue here? Because of the coloring, the blue-green, that's phloem. And if I ask this, that's going to always be xylem. Okay, so just remember those color coordinations there. Okay. Let's see. Before I move on to the organs, one last thing here, and I'll just keep it on this slide for just a second, is that the vascular tissue, as I've mentioned already, allows for increase in height of the plant, right? So they're allowed are able to get taller and taller, so that they can outcompete for space and also for light. Now, one of the things that you have to think about, though, is that a plant can be pretty spindly, and it can grow and grow and grow and grow and grow in length up. The Theophyta, which were your brown algae, did that. I said that some of them can grow about a foot a day, and they are very spindly, but they've got the water and their little air bladders to keep them upright to get the light. Well, these guys don't have that. So when these vascular plants, seedless vascular plants, are growing up and up and up and up, then they're totally at the mercy of gravity, and gravity could easily just kind of pull them down. And so there's two different types of growth then that evolve over time to allow for the plant to grow tall without falling over and succumbing to gravity. And that's going to be primary and secondary growth. So your primary growth, guys, is going to be growth in height. Okay, so the apical meristems that we talked about in the last lecture are here on this plant. So the stems are going to continue to grow up at the apical meristem, and then the plants are going to continue to grow down into the soil at the apical meristems in the roots. So as they grow down deeper to the soil and up in the stems, they also then are going to eventually need what's called secondary growth. And secondary growth, without going into a lot of the detail here, secondary growth is just simply going to be growth in girth. So the plants are going to get much wider so that they can grow tall without falling over. So the vascular tissue allows then for primary and secondary growth. Okay, so hopefully that makes sense. And just a reminder, if it doesn't, go ahead and send me an email and we'll work our way through that material. Okay, we're going to move on and we're going to talk about organs. And last time we said that the bryophytes were tissues without true organs. So here we see true organs. Okay, so this is another derived trait. And we're going to start first with leaves. So you probably are familiar with leaves. I can rest assured that you guys all know what a leaf is. And the leaves, if you want to think of them, they're kind of like little solar panels, right? So you can see these guys right here, and they're spaced beautifully to maximize then capturing sunlight, specific rays of UV radiation, usually in the blue, violet, and the red colors then that are going to allow for photosynthesis to take place. But leaves didn't always look like this, right? Obviously, you can imagine it took many, many years, millions of years, in fact, for leaves to become this size and as advanced as they are here in this picture to the right. So I'm going to go over here and I'm going to look at this here, which is called a microphyll from 2913 in your textbook. These came about 410 million years ago or so. And phyll means leaf. Phyll down here means leaf. Micro obviously means small, and mega is big. But that's kind of a confusing thing here because if you look at this picture and this picture, it's not like these leaves are huge compared to these leaves, but I will show you what's different about the two. So if we go to the first types of leaves to have evolved, your microphylls, these are described as being leaves that have a single unbranched vein. So if I look at the darker green in this picture right here, that's the main vein. And if I were to ask you what is a vein is a vascular bundle made up of the xylem and the phloem. So you've got this xylem and phloem moving through the plant, and every time there's a microphyll, you get one stem of it break, or I shouldn't say stem, but one pass of that vascular tissue through that microphyll and over here as well. If you go to 370 million years ago, this works well, but it's not adequate enough for the plants to come. And so eventually we get megaphylls, where again, the leaf itself can be small, okay, but mega means that there's an increase in the vascular tissue. So we see that there's a branching of the vascular tissue because there's a branching of the leaf tissue. So lots of different kind of finger legs, if you will, coming off of here and then into these little folds and different areas of the megaphyll. This right here is going to increase surface area. So we have a much more efficient system, allowing for the movement of water and mineral nutrients, okay, to be much more efficient than this would over here. Okay, so those are your leaves. Now, your leaves aren't always just going to be for only photosynthesizing. There are leaves that are adapted for other functions, and that's going to be what is called a sporophyll. So let's just look at the term here, guys, and it might be worth noting that these new terms here, if flashcards work for you, that might be a good thing to at least start with so you can just kind of know the roots. So if you remember from the previous slide, I said phyll meant leaf. Sporo, you've seen this before, like on the sporophyte, so this is spore and this is leaf. So anytime you hear me talk about or you read about what are called sporophylls, please understand that those are just leaves that have been modified to have sporangia. Now, if we go back over here, because there's lots of terms, sporangium, the angium, that's holder of. And so if I were to ask you, what is this holding? You should be able to tell me spores. And this is going to be then on the sporophyte stage of the life cycle. Okay, so nevertheless, the sporophylls are going to be found usually in two different kind of formats, either what's called a sorus, singular, or sori, plural, or a strobilus. So if I look at this one over here first, some of the plants that you guys are going to look at today in this lecture and then also in the lab are going to be ferns. And I'm sure there's probably a fair number of you that are familiar with what a fern is. But if you turn over the fern leaf, which is actually called a frond, you're going to see all of these little tiny dots on the underside, right, of some of those fronds or leaves. So if you take this area right here and you blow it up over here, each one of these little clusters, okay, is called a sorus. And if you look at the sori, what's happening here is that there's all of these little kind of round circle things, and these are all multiple sporangia. So each one of these little circles, so this guy, for example, is a single sporangium busy making spores. There's another one doing the same thing, another one here, another one there, another one there. And so you can imagine that if there are this many sporangia in each cluster called a sorus, how many spores do you think that this sporophyte can actually make? And usually in a typical fern life cycle, that's what this plant again is right here, they'll make well over 1 million spores, okay? So that's what one version of a sporophyll would look like. So it's a leaf, in this case specifically a frond, that has these sporangia on the underside. If I go over here, a different type of plant that we're going to look at today, then you've got this little area coming up from the rest of the plant, and it's got this little cone-like structure on it. There's another one there. There's two more over there. So this right here then is equivalent to a sorus, but it doesn't look the same, so they've given it a different name, and that's going to be called a strobilus, singular, strobili, plural. So if you were to take and cut this longitudinally, you're going to see that there are several sporangia in there, making spores. So again, this little guy here called the strobilus has the capacity to make probably I don't know about a million, but thousands and thousands and thousands of spores. Okay, so sporophyll is a modified leaf that has sporangia on it somehow, some way, either by making sori here or by making a strobilus there. Okay, I'll come back and we'll highlight those as we go through the different life cycles of the plants so that we're sure to understand what we're talking about here. Okay, next, seedless vascular plants, then in addition to having leaves, are also going to have stems. And the stem, basically, guys, this is the part of the plant that grows above ground. The stem, the major function for the stem is to position the leaves so that the leaves are going to be able to maximize sunlight at their greatest capacity. It wouldn't make a lot of sense if, for example, you had all of the leaves just growing out of one part of the stem, right? They need to be spread out and probably on both sides here so that we can maximize the capturing of sunlight for photosynthesis. Now, not all stems are going to look the same. Okay, it just depends. This is going to be a woody stem. This is an herbaceous stem, which means that there's no wood. And then at the tips of most stems, you're going to have these areas here that are going to be called the terminal bud. And that's either going to make more leaves or a cone of some sort. In some plants, though, in addition to having their above ground stems, they can have different types of stems, sometimes growing underground. So these right here are all different versions of modified stems, and they are growing underground. So this right here is called a rhizome. So this is the underground stem, and then up from it are going to be shoots with leaves and stems that will grow up through the soil. A plant that you're probably familiar with that has this is going to be ginger. So when you're eating ginger, if you cook with ginger, maybe you make lots of Asian types of dishes, because that is called as an ingredient, that's going to be a rhizome. Okay, over here, this one is a corm. It is a lot, I don't know, more round than the rhizome. This is more elongated. This is kind of like an underground cone, if you will, like think pine cone. And it's got these little tiny root-like structures coming off of it, as does the rhizome, these little root-like structures. It's got a node just like these guys do over here, and it's going to send out leaves, but this is all underground, and it's stem tissue. When you eat a potato, and maybe you leave your potatoes right in your pantry for too long, okay, and they start to smell, and then you're like, oh, what is that? And you go and you find, oh yeah, I had these potatoes in my pantry and never did anything with them. They're starting to kind of decompose, and they've pushed. So at their little eye, if you will, the little eyes, this is what's going to be pushing out of it if this has been in the pantry for too long, and that's actually going to be the stem. And the stem is coming from the tuber, which is actually an underground root. Over here, you've got the stem here, okay, called a bulb, and that's the part that you're eating when you eat something like a red onion. So the point being, guys, is that most stems are above ground, and they're going to be for the positioning of leaves. Vascular tissue has to move through the stems, right? So think of it as a highway to allow for that plumbing system of xylem and phloem to move water and mineral nutrients throughout the plant, in addition to its sugars or photosynthates, but some of them can be underground like these guys there. Okay, and then finally, last derived characteristic before we move on to the respective phyla are going to be roots. So you've got roots here that look very different than just what a typical root ball underground would look like. So these guys here, obviously it's got a massive root ball that's growing underground, and the main job of a typical root that grows underground is to allow for the absorption of water and mineral nutrients. And they're going to take that up through their xylem and then move it up through the plant. But in addition to having the root ball, you can have roots that come out of the ground like this. You can have roots in this is a mangrove forest here, where they're kind of like, they almost look like side shoots coming off, but this is actually root tissue. And then you can have some of them that hang down, okay? And it almost looks like another plant, but it's not. These are going to be aerial roots. And so these roots here that you see that are not growing underground, they are going to be used just for support. So they're more supportive structures than they are basically anything else. They're not necessarily doing any sort of absorbing. They're just going to be there to help support the plant. Okay. So we're going to go ahead and we're going to look at then two phyla that are representing the group called seedless vascular plants. And they are called Lycophyta and then Monolophyta. And so you can see from this table 29.1 from our textbook, this group we already have gone through. I asked you to ignore Anthocerophyta, but we should have gone through Hepatophyta and Bryophyta already, and now we're working down here. So vascular tissue is the big thing here, but we don't have seeds yet. Okay, so seedless vascular plants. We have very few known species of the lycophytes left at about 1,200, but the manilophytes here, we have about 12,000 of those. And this group right here is one that you probably are going to be a little bit familiar with. And then just before I start going through the different groups, just understand that everything that we're going to talk about are homosporous. And so remember that means that the plant is making only one type of spore. That spore gives rise to a bisexual gametophyte. Okay. So phylum Lycophyta. So commonly referred to as the lycophytes. And these, as you can see, they're not huge, okay, at least not anymore. They used to be, but again, I'll talk about that later. But they do have leaves, they do have roots growing underground, and they're going to be much larger and much more sophisticated than any of the bryophytes. Here is another version of the group in Lycophyta. And then here's another one. And we're going to be looking today at something that looks more like this. Okay, typically these guys are epiphytic, which means that they grow on other plants, but they are not parasitic. Huge difference here. If you are considered to be an epiphyte, or if you are epiphytic, that means that they have just simply attached themselves to a plant that is going to grow taller than they will. They are not going to remove any nutrients. They're not going to gain anything except for getting sunlight. So if these plants are able to hitch a ride, if you will, on a taller tree, and that tree puts them up closer than to the sun, so they don't have to compete with other plants on the forest floor, then that's a win-win for them. But again, they don't hurt the tree that they are living on. So some species live that way, while other species are found growing on the forest floor, usually in areas of the tropics, okay? They have dichotomous branching that you can see quite well right over here. So there's your dichotomous branching. And they, again, as I mentioned, they have stems and roots and leaves. Okay. I'm going to go here to talk about then the next characteristics. So this life cycle that I showed at the closer to the beginning of the lecture here. So this is the phylum Lycophyta. This genus is Lycopodium. You do not need to know the genus name, but you do need to know the name of the phylum. Okay, these guys are going to have microphyls. So I have a decent picture of these in the lab intro seed. These small leaves right here, but remember micro, not in that they're small necessarily, but they don't have branched vascular tissue going through. It's just kind of one offshoot. They have these structures called rhizomes, and so underground then are these little kind of tiny root-like structures called rhizomes. They do some absorbing, but they're going to be there primarily for attachment to the substrate to hold the plant in place. So these guys here don't have true roots, or a root system, I should say, like any typical plant that you are probably familiar with. Okay. They are going to look like this in their sporophyte stage, and they look like this then in their gametophyte stage. So if we just take and go through this life cycle here so that you can understand what's going on. I already did it, but we'll do it once more. Remember, this is just going to simply be alternation of generation. Okay, so the sporophyte stage, One of these upshoots here is going to be a leaf that has turned into a reproductive structure. So this is a sporophyll. And this particular type of sporophyll is the cone-like structure called a strobilus. If we were to take and do a longitudinal section of this cone, what you're going to see are little arms, and these little arms, that's called the that's not called, but that's part of the sporophyll. So remember the leaf that has been modified to making sporangia, which are these circles here. So there's one, there's another one, there's another one, and there's another one. And then inside the sporangia that are kind of in place and held by the sporophyll are going to be your spores. Okay, so if we take and we look at this area right here, that's going to be here. So inside this sporangium, meiosis is happening, and we're going to be making haploid spores because this is all diploid tissue since it's part of the sporophyte. But meiosis in here allows for haploid spores. Those spores then are going to be released by the wind currents. They're going to hopefully be moved away from the parent plant. They're going to land in a hospitable environment, and that spore will go through mitosis, and it will make a multicellular haploid gametophyte. This is a bisexual gametophyte, making both egg in the archegonia and sperm in the antheridia. Water is required because you can kind of see from here that there are flagella on the sperm cells. They need to find the egg, swim to it. They fertilize the egg. Now we're back to the diploid stage of the life cycle. So there's your zygote. The zygote is embedded still within the gametophyte tissue. Okay, even though the gametophyte is very small and reduced in size, it's still there. It's going to go through mitosis, and it's going to make then the diploid sporophyte stage. And you can see that the first part of the sporophyte is going to grow up and down, and this little tiny area right here is what's left of the gametophyte, which will basically die off and be nothing here soon, and we get back to the adult sporophyte stage. Okay, one unique feature about this group is has to do with their spores, and their spores have lots of oils inside of them, and their oils are highly flammable. And so they're used by magicians. The spores, you can buy this stuff online on Amazon, and you can basically think of a magician that just like opens their hand, and then a little bit of fire comes out of it. Well, that's because they have these little spores in their hand that they've lit, and the oil just ignites in the spores. They've been used as part of designing the first light bulb. Okay, so they illuminate when they are on fire, if you will. And then some of the first copying machines, the toner, that's the black stuff that goes on the paper to actually make the copy, because they're so black and so small that they were using a collection of these spores to make the first toner in copy machines. Okay, so one last picture that I want to show you. Think of this. If we were to take one of the stems, you can't really make out the stem quite well because it's just filled with all of these microfills, but imagine taking all these little microfills, all the little leaves off of it, and cutting the stem then in half. So that would be a cross section. This is what it's going to look like. So this is a Lycopodium stem in cross section. This is going to be a slide that you're going to be looking at in your lab, so you might want to pay attention to this. So this area right here, again, it looks different but similar in some ways to the first vascular bundle that I showed you. And that vascular bundle has that indicative red and blue tissues. And so as you see here, the phloem is the blue and the xylem is the red. And the xylem is busy moving the water from the bottom of the plant up, and the phloem is busy moving then the photosynthates from source to sink, from where it's made to where it is needed. Okay. Moving on to the second phylum of our seedless vascular plants, and this is going to be the phylum Monilophyta. And these are going to be things like ferns, horsetails, and whisk ferns. I would imagine that you guys are probably most familiar with this group over here. Some of you might recognize this one over here. This over here, probably not. We don't have any of this living anywhere where we are from here in Bakersfield. So this is probably, I would imagine, a plant that's newer to you. Okay, so for the group as a whole, these guys are going to be more closely related to the plants that make the seeds than the previous group, Lycophyta. Okay, so they've evolved after the lycophytes and therefore are going to be more similar to the seed plants that we're going to talk about next lecture. Again, we have got our ferns, our horsetail, and then what are called whisk ferns. And that's going to be this guy right here. Okay, and so you can go read a little bit more detail if you'd like from your book, and this is going to be figure 2914. Okay, so what I'd like to do now is I'd like to just go and talk about these three different examples, and we're going to go ahead and we're going to start with the ferns. So the good news is that all three plants for this group are in the same phylum, Monolophyta. This is a new grouping. It didn't used to be that way, but now they are all grouped together. So a little less that you have to learn. Okay, one thing that I want to point out is that when you're looking at a fern, it has very obvious leaves, right? But the leaves are actually called fronds. So I've given you some space here in your notes to just take down little tidbits of information for each of these groups. So that might be something that you want to write down, is that frond, there it is right there. The frond is the actual name for leaves in ferns and only in ferns. Why they gave it a different name, guys, I don't know. Maybe it's just to confuse us and more terminology that you get to learn. Yay, right? But just know frond means leaf, but only for the ferns. Okay. Note, on the underside of a frond, you're going to have these little guys here. Those are your ciliary, and those, if you have already forgotten, those are going to be your clusters of sporangia. So because I have the sporangia on this particular frond, then this is actually a sporophyll. Okay. This is one example of a plant then that does have megaphylls. So I think I misspoke and I said that all the plants today were going to be microphylls, but I should not have said that. So the lycophytes have the microphylls. These guys are going to have the megaphylls, at least these ferns. So let me show you what I mean by that. So this purple line that you have that starts to branch off, right, as the plant starts to kind of divide into different sections, that's going to be the vascular bundle. So that's going to be, think of it like your vein in your hand, right? But it's not carrying blood like I mentioned before, and it's not veins and arteries, but it is a bundle of the xylem and phloem. But if you look closely, it branches off into even smaller, like little networks of vascular tissue. You can see them. These would kind of be like your capillaries. And so it's highly branched, making it then a megaphyll, not a microphyll. Okay, these guys, again, they're going to be homosporous. So all of the spores that come out of the stauroid are going to all be identical, and they're all going to make a bisexual gametophyte. These are two different gametophytes in this picture, okay? So they are easy to remember because they kind of have like a little heart shape to them. So the fern gametophyte always looks like this. And to just try to give you some perspective, they're probably only 2 to 3 millimeters across. They're going to be found just under the soil surface. And so pretty insignificant as far as what you're going to be able to see. And they only last long enough to make their gametes, and that's it. So again, this over here is the sporophyte, which is the dominant stage of the life cycle. This is the gametophyte. So I'm going to show you this one. I think this one's a little bit easier to see, although exactly what I'm going to say here is the same on this one. But right kind of where you have an invagination on your little heart shape here, you've got these kind of little darker dots, and these are going to be antheridia and archegonia. So there's a mixture of the two different gametangia. So sperm and eggs are made here in this area, all clustered together on the single gametophyte. You're going to have these little rhizoids coming off of it, and they're kind of root-like structures, again, that I had mentioned earlier in the previous slide, and they're basically going to be used just solely for attachment. Okay, these plants overall are quite important to the ecosystem for succession. When you have secondary succession and you're going to get things like your liverworts maybe coming first, then they're going to prepare the soil for things like the ferns. And these guys are going to obviously be taller than your bryophytes, but they're going to start to fill up the landscape. And that's good because what it does is it allows for habitats then to return for small animals and insects. So that the ecosystem there, that community can get up and running once again. Okay, and then this slide over here, this is just kind of interesting, I think, kind of a fun name. So when a frond is is just getting ready to open, so it's developing. It makes this structure right here that's called a fiddle head, and so it looks just like a fiddle head. And these little coiled structures right here are the immature fronds. So this right here once looked like this. They will basically unwind themselves and spread out, making them these mature fronds. These guys are edible, some species, so please don't go and just start tasting fiddleheads, okay? Some species of these, they are a delicacy, especially over on the East Coast. And some of the species here are really important because in addition to helping with succession, a lot of them house those nitrogen-fixing bacteria that we've already spoken of. Okay. Here is the life cycle of a fern. This is straight out of your textbook. I'm going to highlight only a few things here, but once again, guys, this is just simply alternation of generations with the sporophyte stage as the dominant stage of the life cycle. Here's your bisexual gametophyte that I was talking about. The antheridia and the archegonia. The sperm swim to the egg. You get the zygote that's diploid. It goes through and it makes then a new sporophyte coming off of the gametophyte just temporarily. And then eventually we end up with our mature sporophyte. Okay, so there's going to be a couple of terms, I believe, in your lab that I just want to make sure that you are familiar with. So one of those terms that sometimes comes up or that you might read about is this thing called a petiole. And the petiole then, guys, is just simply this little attachment. So the petiole is where the leaflet or the leaf attaches to the stem. And then this here is a rhizome. And the rhizome is going to be an underground stem, like the ginger, that grows to allow then for attachment of the sporophyte with little rhizoids coming off of it then for absorption. When this plant is ready to release its spores, so if we look at the underside of this frond, right, i.e. the sporophyll here, little sori. When I take one of these little sori and I look at them magnified, here is the sorus, the single sorus, and these little things are the individual sporangia. Sporangium is singular, that's why they have that there. And if we look at one of those and we look at it up close, then it's going to look like this. And it's going to kind of have this little tiny mohawk on it called an annulus. And actually, what I'm going to do here, guys, is I want to go show you that in a previous slide. So give me just a second. My computer is slow to respond here. Okay, without making you hopefully super dizzy, let's go back up here so I can show you a close-up of what I'm talking about. So if you look at, let me expand this so we can see it. Maybe. Okay, we're back in action. So that sorus there, a cluster of sporangia. Each of those little sporangia, there's a little mohawk. It's that little kind of brown structure that you can see on each of the round yellow structures. So if we go back then, so let's minimize this. Okay, and we're back to our fern life cycle. So there's that little mohawk area, and that is called the annulus. And so why I'm pointing the annulus out is because what this is going to do, guys, is this is going to act kind of like a spring. So you see how there's this crease right here. So the sporangium, the head of the sporangium with the annulus is going to open up. It's a little container, right? And inside of there, meiosis is occurring to make those haploid spores. Now, this only wants to open when those spores are mature and ready to be released, and it doesn't want to release all of them at the same time. Again, it's kind of likened to a salt shaker, a little bit at a time. And so think about this annulus as a spring. It's going to kind of pop the head back to look like this, releasing some spores. It'll close and then pop back open again at a different time, releasing more spores. So those spores then are going to go be carried by wind, grow into the young gametophyte that is bisexual, and then we already talked about that story. So the petiole, the rhizome, and the annulus are things that are in the lab that I just wanted to draw your attention to. And then this one last term right here, indusium. So they're not showing an indusium per se on a sorus, but what an indusium would be is it kind of would look like this area right here, this kind of little black thing right here. It's a covering. The indusium is a covering over the individual sori. And that's going to protect and care for the sori as they mature. So the indusium, this umbrella-like cover right here, will basically dwindle down and fall off when the spores are close to being mature and ready to be released. Okay, so I'll show you a picture in the lab intro of an indusium, but I just wanted to introduce you to that term right here. So those might be terms that are worthwhile writing down in the notes, so you don't have to maybe go back to the lecture to find them. Petiole, rhizome, indusium, and then the annulus. Okay, the last two representatives then for the group of Monolophyta, we're going to have horsetail, and horsetail is the common name for these plants. Equisetum is the genus that you will not need to know. And this is what they look like. So maybe you've seen clusters of these guys. We have some actually growing in the planter just outside of our lab that look just like this. They can be, I don't know, maybe 2 to 3 feet tall. They have separate vegetative, which are sterile, and reproductive or fertile stems that look like this that I will highlight in the lab. This one right here is the is the vegetative or sterile stem. This one right here with the cone, that's the strobilus, and you should know that by now. That's the reproductive structure, right? So lots of sporangia inside there making haploid spores. They do have leaves, although the leaves here are not obvious, but they have little rings of leaves. So they look like these little teeth or these little triangles. Those are actually the leaves, okay? These guys are very rare. We have only one genus left. The Equisetum is all that exists anymore. They are found in areas that are usually pretty swampy, and so little to no water flow, and then therefore very little gas exchange and little oxygen that the plants need. And so in order to survive, what they've done is, if you could take one of these and cut it at two ends, so cut it there and then cut it there, it's going to look like a straw. It's totally hollow inside. So if I were to take and cut this in a cross section and look at it from the top down, this is what I'm going to see over here. So the plant here, the Equisetum or the horsetail, is filled with all of these air canals, a massive air canal in the center. So that would be like the straw area. And then within the tissue here, this green tissue that you can see, you're going to have many smaller air canals going around the main air canal. Okay. And then the last group that we're going to be looking at for the manilophytes are going to be the whisk ferns or the Psilotum. And again, that's going to be a plant that is not indigenous to our area. So this is probably something that I would imagine you're not quite familiar with. But a pretty cool plant in that it is made 100% of stem tissue. There are no leaves on this plant and there are no roots. So the above ground is all stem. The below ground is all stem. Very obvious dichotomous branching that you can see there, okay? And all of these little yellow nubs that you can see all along the stems are these guys right here. There's one there, and one there. And these are called synangia. So these look different. It's not the same thing as a sorus or a strobilus, but it's a counterpart to both of those. And we have then one sporangium, another sporangium, and then a third sporangium. And so you can see that they are all fused together, especially in this one. There's one sporangium, a second one, and a second one, making up a synangium or synangia plural. And when they open, they are just packed, filled with mature spores. So thousands upon thousands upon thousands of spores there. Okay, if we were to take a stem and cross it here to make a cross section, here's the outer green part right here. And then this is going to be the Singer vascular bundle. And by now you should be able to tell me that the red is xylem and the blue is phloem. Okay. Okay. And then this again will be something that will be highlighted in your lab, and you'll see that in the lab intro. Okay. To finish up then, why are these plants important and what sort of an impact have they had on the environment? So if you can go back and think about the first picture that I showed in the lecture, right, which was the swampy area with the massive plants that were growing, 6 feet in width, 240 feet tall. Okay, not that they were all that big, but a lot of them got to be that size. And as they had this massive amount of increased growth, then they also massively increased their rates of photosynthesis. In order to keep yourself alive, if you're that big, you have to be pumping out sugars constantly. Okay, and in order for photosynthesis to occur, they have to take carbon dioxide from the atmosphere. And what that ended up doing was, because they were so successful, this is about 350 million years ago to 300 million years ago, they sucked so much carbon dioxide out of the atmosphere that decreased, right, the greenhouse gases, which then they believe probably led to global cooling. And when I mean global cooling, I mean basically an ice age, okay? So they were too successful for themselves. They could not survive then in this ice age, in this very cool environment that they had created because of their increase in photosynthesis. And so they died. And these giant trees, right, 6 feet by 240 feet then, that are living, just die and they fall over. And you know that decomposition is going to come in and take care of these, but because they're in a very swampy area, the rate of decomposition can take years and years and years. And so as they have fallen down in these wet, swampy areas, they're not decomposed rapidly. Other things that die fall on top of them, and then other stuff falls on top of that. And basically, you just get these layers and layers and layers of these carbon-rich plants, right, that become peat and coal because they're without oxygen, and they have tons and tons and tons of pressure on top of them, then becoming or making them become coal. And so what's interesting about this is that we now are taking advantage of that, because what are we going in? What are we looking for? Especially lots of places around here in Kern County, we're looking for not necessarily coal, but for petroleum products, for our things like petroleum and oil, so that we can make our gasolines. And so a lot of those carbon compounds then are due to the fact that these plants from millions of years ago died, decomposed, or didn't decompose completely, had a bunch of weight on top of them, turning them into these petroleum products. And so, again, another reason to go and thank plants, right? Because now we have oils and petroleum products that came from these plants. Okay, so that is all that we have then for this lecture. And the next couple of lectures, we're going to finish up, and we're going to be talking about seed plants. Okay, that is all for now. Thank you very much for listening. Have a great day.